So, a warm welcome everyone. I'm Dr. Chandrasekhar, working in the Department of Biotechnology. So, I'll be dealing with the course name Cell Biology, and its course code is 23BT1101. It is a FED course. So, today our topic is classification of organisms. So we all know that there are wide varieties of organisms in this world, and classifying these organisms into groups and subgroups is not an easy task. So scientists have put forward so many theories and so many classifications, like 2-kingdom classification, 3-kingdom classification, as well as 5-kingdom classification, and so many other classifications. Among those, 5-kingdom classification is one of the most widely accepted classification in classifying the organisms present in this world, right? So before we go into or dive into the classifications, we should understand what are the taxonomical ranks for doing the classification, right? So if we have a look at this taxonomical ranks, domain has highest taxonomical rank, followed by kingdom, followed by phylum, followed by class, followed by order, followed by family, followed by genus, and the last one is the species. Species has the lowest taxonomical rank in any classification, right? And the domain has the highest taxonomical rank, right? So domains, there are three major domains of life. Domain 1 is called as archaea, and domain 2 is called as bacteria, and domain 3 is called as eukarya, right? Three major domains of life: archaea, bacteria, and eukarya, right? So in domain archaea, they are all ancient bacteria which are present. So all the ancient bacteria have been kept under the domain archaea, and bacteria under the domain bacteria, all the current generation or current organisms or eubacteria are placed under the domain bacteria. Coming to the eukarya, eukarya means all the other remaining eukaryotic organisms are placed under the domain eukarya. Okay, so basically three major domains of life. First one is called archaea, second one bacteria, and third one eukarya. Domain archaea has all the organisms which are ancient, very old organisms, microorganisms, and domain bacteria has the eubacteria, are also called as true bacteria, and domain eukarya has all the other eukaryotic organisms, right? So I hope you all know the difference between a prokaryote and an eukaryote. So prokaryotes are unicellular microorganisms which does not have a definite nucleus inside the cell. So prokaryotic organisms do not have a definite nucleus and any other double membrane bound structures or internal organelles present inside the cell. Whereas coming to eukaryotes, eukaryotes are the organisms which are multicellular in nature, and they have a definite nucleus inside the cell as well as all the other intracellular organelles inside the cell. So we'll have a look at the differences between prokaryotes and eukaryotes in the next slide. So for time being, remember that all the prokaryotes are present in your archaea as well as the bacteria. So these are unicellular microorganisms. Right. So let's move on to the 5-kingdom classifications. So in the 5-kingdom classifications, so as I said before, domain has the highest taxonomical rank, and domain consists of three major domains of life: archaea, bacteria, and eukarya. Next one what we have is the kingdom, right? So one of the most widely accepted classification in kingdoms is five kingdom classification by Robert H. Whittaker. He proposed in 1969, and this five kingdom classification is mainly based on the anatomy of the microorganisms or anatomy of the organisms, morphology, embryology, as well as the cell structure of the organisms. He had a look at all these components, and then he has segregated all the organisms into five different kingdoms. So what are the five kingdoms according to the Robert H. Whittaker? So the first one is called as monerans, and the second one is called as protistans, and the third one is called as plantae or plants, fourth one fungi, and the last one is called as animals, right? So five major kingdoms in Robert H. Whittaker's classifications. Now let's have a look at what are there in each kingdom, right? So coming to monerans, all the prokaryotic organisms are placed in the kingdom monera. All the unicellular microorganisms are present in the kingdom monera. Your bacteria and archaebacteria comes under monerans. Coming to protistans. So protistans are multicellular eukaryotic organisms, or some cases they are unicellular organisms as well, right? So here in case of protista, you have wide varieties of microorganisms, including your slime molds, green algae, flagellates, amoebas, red algae, brown algae, dinoflagellates, diatoms. These are all different varieties of organisms which you generally see in sea, right? These are all present in the kingdom protista, second kingdom. So the third kingdom is plantae. We all know about plantae, right? Plantae, all plants present in this world are kept in this kingdom plantae. This plantae includes your gymnosperms, angiosperms, mosses, ferns, all these kinds of plants in this world are kept in this kingdom plantae. And fungi, another kingdom. In the fungi, you have molds, yeast, fungi, all the other fungi are placed in the kingdom fungi. And the last kingdom, according to the Robert H. Whittaker classification, is animalia or animals. All the animals, chordates, non-chordates, echinodermates, arthropods, annelids, mollusks, flatworms, sponges, all these organisms are kept under the kingdom, the animalia, right? So I hope you are clear. You have a clear understanding between kingdom and domain. Domain has the highest taxonomical rank, followed by kingdom. Domains, there are three major domains of life: archaea, bacteria, and eukarya. Only three domains. And kingdoms classifications, so many varieties of kingdom classifications are there, but one major classification among them is five kingdom classification. In the five kingdom classifications, monerans, protistans, plantae, fungi, and animalia constitute the five major kingdoms, right? So let's move on. Right. So let's have a look at the differences between prokaryotes and eukaryotes. Just now I discussed what is a prokaryote and what is an eukaryote. Unicellular organisms which do not have a definite nucleus inside the cell is called a prokaryotic organism, and multicellular organisms with a definite nucleus in its structure is called as eukaryotic organisms. Prokaryotic organisms example, bacteria and archaea, as you've seen in the earlier slide. And eukaryotic examples, protista, fungi, plantae, and animal, all under eukaryotic. Multicellular organisms are kept under eukaryote. So what are the other major differences between these prokaryotic and eukaryotes? So type of nucleus, as I said, in case of prokaryotes, there is no true nuclei. So the genetic material that is there inside the prokaryotic cell is called as nucleoid. Whereas in case of eukaryotes, you have a true nucleus with a double membrane bound structure and DNA and chromosomes present inside the eukaryotic cell, right? So the genetic material in a eukaryotic cell is in the form of DNA, which will be condensed, highly condensed to form chromosomes, and they helps in separation during the mitosis as well as meiosis. So next major difference in case of DNA, in case of prokaryotic organisms, you have a circular DNA, whereas in case of your eukaryotics, you have a linear DNA, which is wrapped around by a specialized proteins called histone proteins, right? Histones are the proteins which have high affinity for DNA. They go and bind to the DNA and helps in wrapping around the DNA and coiling the DNA to form chromosomes. Next one, ribosomal composition, whereas in case of prokaryotes, we have 70S ribosomes, and whereas in case of eukaryotes, we have 80S type of ribosomes. So we'll discuss more about ribosomes in coming sessions. And cell movement is happening in case of prokaryotes with the help of external appendages that are present to the unicellular organism, that is flagella as well as cilia, which helps in the locomotion or movement of these organisms. Whereas in case of eukaryotes, not only flagella and cilia, lamellipodia, filipodia, these are all the structures which helps in locomotion of the eukaryotic cell. Mitochondria. So as I said before, there are no major internal organelles in case of prokaryotes. So there is no mitochondria, whereas in case of eukaryotes, there are several hundreds to thousands of mitochondria present inside the cell. And chloroplast, no chloroplast, whereas in eukaryotes, there are chloroplasts in plants as well as in algae, right? Next one, organization, how the cells are being organized. In case of prokaryotes, as I said, there is only one cell or single cell, so there is no specific organization since it's a single celled organism. And whereas in case of eukaryotes, these cells will aggregate to form colonies in case of some organisms, and in case of higher organisms, it is multicellular. There will be bonding between two cells, there will be attachment between two cells with the help of integrins and other molecules, cell binding molecules, and these cells will form tissues, and tissues will form organs, and organ will be constituting the entire organism. Okay, that is in case of higher organisms, multicellular organisms, whereas in sponges and other multicellular organisms, there will be simple assembly of cells into colonies. Okay, that also comes under your eukaryotic multicellular organization. Coming to the next important difference between prokaryotes and eukaryotes in case of cell division. So in case of prokaryotes, you can see only one type of cell division, that is binary fission. Simplest type of division, where a cell constriction will form in the middle bacterial cell, and the cell will separate into two individual cells, right? Whereas in case of eukaryotes, you have mitosis and you have meiosis. There are two different types of cell divisions that's happening in case of eukaryotes. In case of mitosis, where one cell will divide into two exactly similar cells, whereas in case of meiosis, one cell will divide into four cells which are not similar to the parent cell. Okay, so in case of mitosis, it is called identical division, and whereas meiosis is called as reductional division. We'll discuss more about mitosis and meiosis, how the separation happens, how the cell division happens, what are the phases. We are going to discuss in detail in coming sessions. Coming to the membrane, so as I discussed, there are no membrane bound internal organelles, but in case of prokaryote, it has a cell membrane on top. all the bacterial cells will have a cell membrane, and on top of the cell membrane there is a cell wall as well. So this cell wall is called a peptidoglycan layer. The cell wall of bacterial cells is made up of a peptidoglycan layer. Below the peptidoglycan layer, we have the cell membrane. Okay. Whereas in case of eukaryotes, some types of organisms will have cell wall, like a plant will have cell wall made up of cellulose, hemicellulose, and pectin. And whereas eukaryotes, some eukaryotes like animals which do not have a definite cell wall, right? That's one major difference. But all the organisms will have a cell membrane as well as intracellular organelles like mitochondria, chloroplast. All these are again bound by a membrane, double layer membranes. Some are double layered membranes, some are uni-layered membrane. Like they take the case of mitochondria, take the case of Golgi complex and the plasmic reticulum. These are all the double layered membrane bound organelles, whereas your lysosomes are uni-layered membrane organelles. Right? So that's some of the major differences between a prokaryote and a eukaryotic cell. Let's move on. So let's have a look at what are the cellular appendages. Okay. So let's have a look at what are the structural components of prokaryotic cell. Right. So we'll go one by one. First we'll have a look at what are the structural components of a prokaryotic cell. Then we'll move on to the structural components of a eukaryotic cell. So structural component of a prokaryotic cell can be divided into cellular appendages, cellular envelope, and intracellular structures or internal structures. So structural cellular appendages means the attachments that are present to the cell are called as cellular appendages. So what are the appendages here? We can see we have the flagella, we have the pili, we have the cilia. So these are all the different types of appendages that can be present for a bacterial cell. Most of these appendages like a flagella or cilia helps in locomotion or movement, whereas in case of your pili, which helps in transfer of genetic material from one bacteria to the other bacteria. And coming to the cellular envelope. Cellular envelope means the covering of the organism. Since it's a prokaryotic, it has single cell, it has a cell wall. As I said, cell wall in a bacterial cell is made up of a peptidoglycan layer, whereas cell membrane is made up of lipids and proteins. Cell wall will be there on top. On below the cell wall, what you see is the cell membrane. So you have a specialized layer called glycocalyx on top of the peptidoglycan layer and cell wall, which is more diffused layer which is present on bacterial cell. This is also called as slime layer or glycocalyx, which is present on top of the peptidoglycan layer. So total cellular envelope of bacteria constitute slime layer on top or glycocalyx on top, followed by cell wall, which is made up of peptidoglycan layer, and towards interior of the cell we have the cell membrane, right? So below the cell membrane, what you see is the cytoplasm, right? So in the cytoplasm, you see all these intracellular organelles, right? What are the intracellular organelles that are there in the prokaryotic cell? We have the chromatin bodies, which is nothing but the DNA or the genetic material of the prokaryotic cell. We have the plasmid, which is nothing but extra DNA, extra chromosomal DNA that is there inside the bacteria is called a plasmid. And we have the ribosomes. Ribosomes are the factories for synthesizing the proteins, and we have the endospores. So endospores are the structures which are generally formed during unfavorable conditions, when the temperature is too high or the environmental conditions are bad. When the organisms are unable to survive, they will go into the endospores, form the endospores, and they will survive during these tough conditions by creating these endospores, right? So these are some of the intracellular structures, and appendages and cellular envelope of a prokaryotic cell. So let's move on to the eukaryotic cell, right? So we have seen in the earlier slide prokaryotic cell and components of the prokaryotic cell. Now what you see here is a schematic representation of an eukaryotic cell. Okay, so this is an animal cell, as it's clearly indicated there is no cell wall. Since there is no cell wall, this is an animal cell. The outer layer, what you see, the orange color one, is nothing but the cell membrane, which is, and you have all the major intracellular organelles inside the cell. So what you see here is the nucleus, very prominent nucleus. You can see here in case of eukaryotic cell, around nuclei we have the DNA inside the nucleus, and we have the nucleolus again inside the nucleus, right? So this is in the form of chromatin. You cannot see chromosomes here in the nucleus. Chromosomes are observed only during the cell division. Whenever the separation need to happen after the replication is happened, then only the chromosomes will form. Otherwise, it will be in the form of chromatin. Genetic material will be always in the form of chromatin, right? So after that, our nucleus is a double membrane bound structure, and beyond the nucleus, outside to the nucleus, with the continuation to the nuclear membrane, what you see here is the endoplasmic reticulum. So these are reticular structures, which means net-like structures or mesh-like structures, which are studded with ribosomes. These dots, what you see here, is ribosomes. Ribosomes present on top of the endoplasmic reticulum. For the major function of the ribosome is synthesizing proteins, or they act as a protein factories. So what you see here, which does not have ribosome, is called as smooth endoplasmic reticulum. Okay, so two types of ERs, rough ER and smooth ER. Rough ER is the one which is studded with ribosomes, and smooth endoplasmic reticulum are the one which do not have ribosomes on top. So there is no protein synthesis, but instead they helps in production of lipids and steroid and other lipid metabolisms happens in case of smooth endoplasmic reticulums, right? So you have other major internal organelles like mitochondria. You have the Golgi complex, you have the vacuole, you have the microtubules and microfilaments, which are nothing but the skeleton of the cell, also called as cytoskeleton of the cell, which gives a rigid shape to the cells. A rigid shape to the cell is given by the cytoskeleton, which is constituting microfilaments and microtubules. You have the cytoplasm, and you have the, whereas if it is a plant cell, you can see chloroplast as well. Since it's an animal cell, you do not find chloroplast in the animal cells, and this is bound by a doubled bound cell membrane. So these are the major intracellular organelles present inside a eukaryotic cell, right? So so in this class, this is a summary class or introduction class. We discussed about the basics of organization or stratification of organisms, that is classification of organisms. Then we have discussed about the difference, what is a prokaryote and what is an eukaryote, and the differences between prokaryotes and eukaryotes. And then we have discussed about the what are the components of component parts of a prokaryotic cell, right? How does a prokaryotic cell look like? What are the structural components of a prokaryotic cell? What is its major intracellular components, and what is the envelope made up of? We have had a look at all these things, and we had a look at the structural composition of eukaryotic cell. This is a eukaryotic cell. How does it look like? What is the schematic representation, and what are the major types of intracellular organelles that are present inside the eukaryotic cell? Okay, I hope you understood this session. We'll wind up here and we'll continue in the next session. Thank you.